Today's topic is about mode of action of enzyme, means how exactly this enzyme is working in our body with the which mode of action that we will look in this video. So first, this enzyme is working based on that lowering of activation energy. Activation energy is defined as the energy required to convert all molecule of substrate from the ground state to the transition state. Now, substrate are remaining in an energy through and are to be placed at a higher energy level, whereupon spontaneous degradation of the substrate to the product can occur. So simple substrate is converted into the product, but before that substrate is getting energy and getting a put it over a high energy level, and from that transition state it is spontaneously converted into the product. So for example, activation energy for acid hydrolysis means in the presence of acid, hydrolysis of sucrose, that is disaccharide. So sucrose will be hydrolyzed to the glucose and fructose is possible by providing 26,000 calorie per mole, while the activation energy is only 9,000 calorie per mole when the same molecule is hydrolyzed in the presence of enzyme sucrase. So that is advantage of the sucrase. So in the presence of this sucrase enzyme, requirement of activation energy will be decreased. So that is one of the important mode of action for the enzyme. So here also same way explain lactose, that is also disaccharide which is made up of glucose and galactose. So lactose can be hydrolyzed with the present without enzyme, and for that activation energy requirement will be higher, but same reaction will be done in the presence of enzyme, that is lactase. So requirement of activation energy will be decreased. So that is important mode of action. So lowering of activation energy. Another example, that is glucose is converted into glucose 6 phosphate in the presence of ATP, but without enzyme that requirement of activation energy is high, but in the presence of enzyme the requirement of energy will be decreased. So that is giving advantage in various biological reaction inside our body. Another mode of action, that is acid base catalysis. So in the active site or active center of an enzyme, a number of amino acid chains can act as a proton donor or acceptor. Proton means simple H plus molecule. So that will be either accepted or donated based on that acid base catalysis. This group can be precisely positioned in an enzyme active site or center to allow proton transfer. So histidine at the active site of an enzyme can gain or lose proton. So that we will look in the picture. First we will look histidine mediated acid base exchange is due to its closer pK value to the physiological pH. Simple, most of the active centers majority are getting that histidine molecule because this histidine that can easily take part in a acid base exchange. Not only histidine, so many other amino acid are also taking part in this acid base exchange, that also we will look in the next table. So this histidine is utilized, basically purpose the pK value is near to the physiological pH. So here a table is shown, that is amino acid residue. Here glutamic acid and aspartic acid are basically they are the acidic amino acid. Same way basic amino acid like lysine and arginine, they are involved in a donating as well as accepting the proton based on the requirement of the or activity of the enzyme. Same way cysteine, histidine, serine and tyrosine, that all can also work as a accepting as well as donating of the proton molecule. So that is important mode of action of this enzyme, acid base catalysis. Same way covalent catalysis, a transient covalent bond is formed between the enzyme and the substrate. A nucleophilic, that is negatively charged, or electrophilic, that is called a positively charged group of enzyme attacks the substrate. So that is called a covalent catalysis. Same as a basically active site or active center where substrate is attached over the enzyme, and that is making a one pocket or socket for the attachment over the enzyme. So that is making a negatively charged group or of amino acid are placed together for the activity of enzyme, or sometimes positively charged enzyme are paired together. Same way serine proteases. So they are enzymes with a serine residue at the active site, and most of the proteolytic enzymes belonging to this group. So trypsin, chymotrypsin, clotting factor, they are basically serine proteases which are containing serine amino acid at their active site or active center. Another important example, that is action of chymotrypsin. It is a combination of covalent as well as acid base catalysis. Covalent means there is a attachment or nucleophilic or electrophilic socket is generated. Same way over that socket acid base catalysis means exchange of proton or proton is occurring. So The peptide bond is hydrolyzed through the acid catalyzed nucleophilic attack utilizing the serine 195 position over that position that serine molecule is present over the enzyme. The enzyme substrate complex is formed by the binding of an aromatic amino acid residue with the hydrophilic pocket on the active site on the enzyme. Basically, this chymotrypsin is towards the metabolism of aromatic amino acid like phenylalanine and tyrosine. First, it will attach over this particular hydrophobic Felix pocket of the enzyme, and over that it will attach, and over that attachment that acid base catalysis will occur. So a covalent acyl enzyme intermediate is formed, and finally cleavage of the peptide bond over that protein will occur. So serine, which is present over 195 position, histidine over 57th position, aspartate over 102 position are basically working as a catalytic group for the activity of the chymotrypsin. So in the picture you can see that aspartic acid 102, histidine 57 and serine 195. So these three are the number of the particular amino acid present over this chymotrypsin. Over that particular protein is attached. Mostly this aromatic amino acid is metabolized by this chymotrypsin. So this peptide bond, that is CONH, and that will be broken down by the activity of this chymotrypsin, which is containing over the active center aspartic acid, histidine and serine molecule. So finally this peptide bond will be broken down in the presence of this particular active center or active site of the enzyme chymotrypsin. Same way, catalysis by proximity. For molecules to react, they must come within the bond forming distance of one another. So when an enzyme binds substrate molecule in an active site, it creates a region of high local substrate concentration. So once there is attachment, then acid base catalysis or covalent catalysis will occur. So basically proximity is needed for the reaction of the enzyme. So this environment also orients the substrate molecule to interact, resulting in rate exchange enhancement of at least 1,000-fold. So once there is an attachment, there are so many minor changes is occurring, and that is favoring for the making of product molecule by that enzyme. So that is catalysis by the proximity. So these all are the basic mode of action for the activity of the enzyme.